Hi all, today I'm talking about a network framework to explore phylogenetic structure in genomic data. So phylogenetic analysis often produce a large data set of competing trees. Usually it is not useful to report the whole set directly. So we need to summarize some interesting information from the large data set, such as hybridization, etc. And also we are interested in identifying those systematic errors during the analytical process. But the current existing methods have some shortcomings. One approach, for example, calculate consensus tree is often used to summarize the tree conflicts. However, it potentially discards information concerning competing trees. Another approach is to project the tree topologies into lower dimensional Euclidean space. However, this method alone may be difficult to interpret. Recently, clustering methods have been used to understand phylogenetic data, but they are based on the distance connectivity, which often requires large computation, and they only consider about non-negative links. So our approach is to apply a graph-based method. Two types of networks are constructed. One is tree topology-based networks with trees as nodes and their similarities as the links. And another one is bipartition-based network with unique bipartitions as nodes and their covariance values as the links. In particular, the second network contains both positive and negative links in it. The positive link means that two bipartitions will tend to appear together, while negative link means that if one bipartition appears, the other tend not to appear at the same time. We then apply the community detection methods on the network to discover different sets of nodes with dense internal connections. So for the first network, those trees with high similarities will be identified together, and for the second network, those bipartitions which have a high probability to appear together will be discovered. Now let's consider about a data set with 5 yeast species and the 106 loci. So 106 gene trees are reconstructed using maximum parsimony approach. The formal analysis shows that the two top candidate species trees are tree A and B shown here. The only difference between these two trees are the placement of scud, which flags hybridization as a possibility. To understand the relationship among gene trees, pairwise RF distance are calculated and the affinity matrix are obtained through the reciprocal of the distances. We then applied community detections and discovered 11 communities. Consensus trees are then calculated for each community, and we found that the consensus trees from the largest two communities are able to discover the two top candidate species trees. In addition, we have several other consensus trees here. They may explain either by the systematic bias imposed by the analytical factors or by the action of biological factors. So more considerations are required. In the next example, we are using the same data set, but our focus shifts to the bipartitions. In particular, we calculated the covariance matrix based on the presence or absence of the bipartitions, and then applied the community detection method. We have some bipartitions which appears either in all trees or only a few number of trees. So those are the singletons showed on the left figure. And also we have two communities with bipartitions which are highly support the species trees. So everything I've talked about is implemented in the software called Treescaper, which is available on GitHub. So that's all. Thanks for listening.